Hello, thank you for your interest. Today I'm going to talk about our work, a hybrid model of genome sequence and chromatin structure for non-coding mutation effect prediction. First, most disease-related single-nucleotide polymorphisms are non-coding genomic variants. The SOTA model embeds the genome sequence using the deep learning model and predicts epigenomic events, and then use the change of the epigenomic events to assess the impact of the non-coding mutation effects. Here we propose a new method which uses both 1D genome sequence and 3D chromatin structure to predict the epigenomic events of a given genome region. Before talking about the model, we want to prove that it's an invalid idea. We assume that the genome region have different genome sequence but similar epigenomic events is because they are closer in 3D. So we define the low sequence high label similarity pairs by comparing the cumulative percentage of the sequence similarity and the label similarity. If the margin is larger enough, then we believe there is a low-seq high-label pair. Here we use the cell line-specific epigenomic events only, which means both epigenomic event and chromatin structure are sequenced using the same cell line. We test whether it's closer in 3D by measuring its interaction frequency, which is measured by the HitSeq sequencing data. If interaction frequency is high, then it's closer in 3D. Because the whole genome label dataset is too large, I have randomly picked up 0.1% of the whole set and repeat 50 times. So one-sided test among how many percentage of my 50 trials are significant is shown in the heat map. The chromatin structure from 3 cell lines GM1 to 878, MR90, and K562 are used. Within each cell line, multiple bioreplicates are collected as shown in different rows. Yellow color means always significant among 50 trials. Dark color means always insignificant. The x-axis moving to the right means increasing of the margin between the sequence and label similarity. So only high difference pairs are left on the right. As shown in the heat map, most of the one-sided tests are significant, which support our assumption. And some rest tests become significant when focusing on the pairs with large differences. So we believe it's a good idea to use the chromatin structure to help the epigenomic events prediction. We integrate the embedded 1D genome sequence and 3D chromatin structure. For a given genome region 1 kbp, the genome sequence is embedded using the SOTA model DeepSeq, which can be easily replaced by any other models. The chromatin structure is represented by an interaction frequency matrix from HitSeq sequencing data. The corresponding row of the interaction frequency is used and embedded by the fully connected layer as the 3D structure information. Then the embedded information is concatenated and used to predict the epigenomic events. By incorporating the chromatin structure and the genome sequence, we have beat the SOTA in terms of the AURC and AUPRC, and the higher chromatin structure resolution we use, the better performance we have. At the same time, we find that the cell line-specific epigenomic events benefit more from the chromatin structure, as higher percentage of them are beating the SOTA compared to the general performance. After the model is trained, we focus on the downstream task, non-coding mutation effects prediction. The cell line-specific ECTLs are collected for the cell line GM1 to 878 and MR90. The SOTA and our model's inference results are calculated and used as the input to predict the expression of the EQTLs is increased or decreased. It turns out that our hybrid model has better performance, especially for the significant EQTLs. The EQTLs has significant expression changes. So our model is not only doing better at the test set, but also at the downstream task. We only use one chromatin structure to train the model and make inference, but there are lots of other cell lines and chromatin structure HitSeq data. So we want to know how sensitive our model is to different cell lines. All available HitSeq data using genome assembly HE19 on ENCODE are collected. The same model is used to make inference on the test set using different chromatin structures from different cell lines. AUPRC is used to assess the performance because the test set is imbalanced. Each box plot is showing the performance of each cell line, and each point is for one bioreplicate of HitSeq data. The right horizontal dashed line is for the performance of our model using the matched chromatin structure. used in the training and test. The black dashed line is the performance of the SOTA. The vertical dashed line is showing multiple clusters of the cell lines, either from same each tissue or have similar disease. We can see that most of them can beat the SOTA's performance, and the best performance is achieved by using the common structure from the same cell line as used in the training. So we have shown that the cell line matters, but the model can always achieve good performance when using some other common structures in the inference. So next, we focus on the inference performance for the cell line-specific epigenomic events. As shown in the box plot, common structures from the same cell line has the best performances for the same, and also for if the common structures come from the same tissue or from the similar cell lines, it's slightly worse, but still better than others. And again, making inference using most of the common structures can beat the SOTA, which indicates the generalizability of our model. Then we try to use the graph model to incorporate the common structure and genome sequence. We still have the same conclusion that common structure is helpful and the cell line specific epigenomic events benefit more from the common structures. So the conclusion is integrating the genome sequence and common structures can outperform the genome sequence only model. Epigenomic events prediction accuracy will be improved if they and the common structure are sequenced from the same cell line. After integrating model is trained, using common structure from same or similar cell lines can always have good performances. The reference we used is the DeepSeek proposed by Joe. Thank you for listening.